Hello, my name is Aleksey Maro. Please join us for the live Q&A session on Tuesday, April 13th, 5 to 6 p.m. Eastern Standard Time. Why are we interested in the ethanol concentrations within food that chimpanzees prefer to eat? This all sort of originated with this study in 2002, which developed into the Drunken Monkey hypothesis, which sought to answer the question of why are humans attracted to alcohol? And what is the cause of alcoholism? This hypothesis is, in its essence, evolutionary, the idea being that all of the great apes, as well as our ancestors, consumed more than 50% of fruit in their diet. Now, the role of the frugivore is relatively well understood as seed dispersal. The frugivore gets calories in exchange for dispersing the angiosperm's seeds. And the drunken monkey adds a third player to this relationship in the form of fermentative yeast or bacteria, which provides an antimicrobial defense by making the environment less tolerable to ethanol-intolerant spoilage microbes as a service to the fruit, and in return gets calories and substrate. The fermentative microbes also contribute to the fitness of the angiosperm indirectly by helping to attract frugivores to the fruit via an ethanol-mediated honest signal that provides a clear indication of the existence of calories in an otherwise often very hostile, very difficult environment where having an edge really helps. Fermentative microbes may be vectored onto fruit just by riding on dust motes or vectored by insects. Now, there's a rich amount of background to this study, and I'm not going to have time to go through all this background even remotely, but suffice it to say, there have not been any systematic studies of our nearest common relatives in the fruit that they eat and the level of fermentation of that fruit in the wild. And so what we wanted to know in the study is just the levels of ethanol that are available within chimpanzee consumed fruit, ideally the preferred fruit, and then the question of whether variation in ethanol concentrations can be predicted by empirically quantified ripeness stages or other fruit metrics. So my study took place in East Africa, in the country of Uganda, at Ngogo in Kibale National Park during the fall of 2019. Ficus mucuso fruits asynchronously, and it can fruit multiple times a year, so there's always some Ficus mucuso around, especially given that it's very common in this particular area of the forest. Here's a video of Hicks foraging in a Ficus mucuso tree. He's eating unripe fruit. And here's another chimpanzee eating some of the more ripe figs. And the result is this sort of carpet of leftover bits of fruit, but also some whole fruit, as you can see here. I collected fruit from the ground, and so all the fruit that I had was fruit that was dropped by chimpanzees, either rejected or dropped accidentally. Ficus mucuso is a pretty large fig, and you could see inside there are some small little holes in the individual structures inside of the figs that are the sites where fig wasps lay their eggs. The fig is unripe. The wasp eggs are simultaneously gestating, and then at some point they develop, and then the wasps emerge from a fig, as you can see here. Then once they are gone, the presumption is that the fig is timed with its mutualistic fig wasps, and the seeds develop into full ripeness at a similar pace. I used a couple of methods to assay the fruit for ethanol. Primarily, I used what's called the gas chromatograph. I would homogenize the figs whole and then smear a little bit of the pulp on the bottom of a petri dish, cover it with plastic, and then after a period of equilibration, I would take out the air, the head space between the fruit and the top of the petri dish with the plastic, and I would inject that into the gas chromatograph, which then I calibrated using serial dilution of ethanol standards with each run of fruit. Now, fortunately, the only video that I have of my lab setup is taken during a rainstorm. This is just to say that there were a lot of methodological issues in getting around the humidity. You have to essentially wait for sunny days in order to do analysis. And so for that reason, I also used an infrared spectrometer that I calibrated using some of the gas chromatograph results in order to process more fruit samples, because the infrared spectrometer will work no matter what the humidity is. So I'm going to show you some results. And so here on the y-axis we have ethanol by mass. Along the x-axis here we have a number of fruits, and there's a little bit of extra space for the various ripeness categories. So I used a couple of different metrics, but they're correlated to the unripe, underripe, and ripe stages of fruit development, and there's a couple that are unknown, so they've been left blank. So here we have Ficus mucuso. Now on the left you have these three that are the three different ripeness stages according to the stage of wasp development that I was able to observe by cutting them open in half and examining whether the wasps had emerged or not yet. And then we have another set of results that are as a product of the puncture force using a penetrometer and binning it into these categories. The result is somewhere between 0.4 and 0.6. Up here we have multiple pairwise comparison notation, and in the both ways of measuring fruit ripeness, we see that unripe figs are have significantly more higher ethanol concentrations than ripe figs. And I think that this is because, again, if the chimpanzees are consuming the biggest and the sweetest fruit, they're also consuming fruit potentially with higher ethanol concentrations, or preferentially selecting fruit with higher ethanol concentrations. What I will get as a result, what's going to be dropped, are going to be fruits with lower concentrations. And there's some evidence for that in the data that I don't really have time to get into. For comparison, I also measured a few other fruits. We have another fig species here, separated by wasp and puncture force class as proxies for ripeness. And you can see that the relationship here is as we would predict, with unripe figs having lower concentrations and ripe figs having higher concentrations. But these were collected from two separate crops where the chimpanzees went and foraged there primarily on one day and exhausted the crop. And then I have a couple other fig species here, both ripe. And you can see the overall ethanol concentrations across these various species are all fairly appreciable. I should underscore that I only collected fruit that was intact and undamaged. There were no overripe fruit collected in this study. Now, these concentrations might not seem very high, but we have here estimates from a study done in Kanyawara. You can see that chimpanzees consume anywhere between 6 to 7 kilos of food daily. Now, assuming on average 90% of that is fruit, then we can make this sort of plot where we have the average daily percent ethanol concentrations, how many grams of ethanol will the animal consume. Now we can convert the grams of ethanol into standard drinks to put it more in human terms, where one standard drink, whether it be beer or a shot or wine, is all 14 grams of pure ethanol. Going by these results of the study, chimpanzees are consuming around 1.5 to 2.5 standard drinks daily. So it's the equivalent of a chimpanzee slowly sipping a beer over the course of an entire day. This result, of course, is conservative because, again, these are rejected fruit, so the actual concentrations are likely higher, and this is also a daily average over the course of a year, and so, of course, there are going to be fluctuations. And this exposure to ethanol via fruit applies to any frugivorous or nectarivorous animal, And of course, it would apply to chimpanzees at any field site. And so the important thing then is to be able to get an accurate assessment of the concentrations that are being consumed. And real quick, I just wanted to throw in this teaser figure from the upcoming manuscript of the study, just to show that there's a lot more going on to tease apart as far as the analysis of this data. Future studies that look at fruits that are being primarily foraged in trees will need someone to climb up there and actually sample some of the fruit, and then, if possible, compare it then to fruit being dropped by chimpanzees and see if what is available to them is lower or higher concentrations than what they're dropping. And there's always the genetic work of whether there is evidence of selection in alcoholic dehydrogenase genes that are involved in the metabolism of this ethanol, because even though chimpanzees might be getting 2 standard drinks worth of ethanol, what that ethanol is doing is maybe different from what our bodies do with it. There are many potential impacts of this kind of research, placing alcohol consumption and alcoholism in an evolutionary context, thus relieving the stigma of alcoholism, reframing it as a clinical disease rooted in evolution, as so many others have been shown. And with that, I'd like to acknowledge and thank the Ngogo field directors and researchers, and of course the Ngogo field assistants, without whom a lot of this wouldn't have been possible. Thank you again for listening, and don't forget to join us Tuesday, April 13th, 5 to 6 p.m. Eastern Standard Time.